somehow we already been studied during our bachelor levels. So I strongly believe this biotechnology has a very big role, especially when it comes under the bioengineering, because we also study the, like when we the what is the artificial intelligence. Normally the images or the research data we got, we do some prediction, and back with the back prediction we just got some knowledge about, this is the basically the prediction based study, but this prediction comes after the experiments, different kind of experiments. In the biomedical also, engineering also includes the biomaterial study, biomechanics, and all these are somehow related to the experimentally, the field of biotechnology. And if you now, in the material science, normally we are now focusing on the developing the various kind of biomaterials for the drug delivery applications, and also the artificial organs, and also like the tissue culture application. But what is this material? Whenever we develop this material, we have to do the biological experiments to know how it is compatible, or how it is good for the application in the biological field. And also when we do some experimentally, it is very expensive to do any experiment, but this due to this computational knowledge, we can do some simulation, and further we can know how this is feasible, and how what kind of results we can get, and from there we can get some idea, and from this small knowledge we can further do some experiments. So this is also comes and somehow we have already studied this when we already performed our graduation in Btec and Mtech biotechnology. So I think this biotechnology is very big and very important subject in the bioengineering. So now this is if you see my example, now currently I am a research fellow in National Cerebral and Cardiovascular Center, Japan. Basically I in my research area, we are doing two kind of study. One is the development of MRI agent using some material for the live cell imaging, and also after that we do the in animal, we apply it in the animal model and see how it is working. And another study is like we are doing some developing some navigator molecule of by synthesizing some protein which have a target to the liver cell, and we captured the protein which is present in the blood level, and we just do the we just means we just captured this protein from the bloodstream and just regulate it to the liver, so that this protein concentration can be reduced in the blood level. So this is both in this both case, I basically I have to use this biotechnology knowledge, and that I got from my basic biotechnology field. So I think this is very important for like doing the experiment and also doing the research in bioengineering field. And also now when I have done my PhD, I developed some kind of material for the drug delivery system. Like we prepare this kind of small micelle and we encapsulate the drug, and from this material, and after encapsulation we just try to do the means like the encapsulation of the drug, and also further we studied how this is the release profile and all these things. That means here I basically my basic background, my basic research is based on the applied chemistry, but in all the time I have to think about how it is good compatible with the biological system. So now that against the I have to perform the biological study, which like the biocompatibility study, and also the cellular imaging and the cell attachment study, and every knowledge I had got from my uh, base my fundamental knowledge which I got from my biotechnology background. So I think this is very important to continue the research in the bioengineering field. and also this drug release profile, like after encapsulation of the drug, we always think about how this drug release is going on in the physiological environment. And that is also, this is not the in the inside the body, but we always perform this kind of experiment in the physiological environment. So this is also means for this study and how the kinetics is the drug release kinetics. So all is very much means correlated, and the knowledge is very much important, which I got from my basic study time, that is in the biotechnology Btec level. And also there here I perform some kind of modification of liposome. This liposome is basically encapsulate the drug and for the further release in the biological environment. So how this liposome can be stabilized and can be good compatible with the biological system. This is also like the example of the combination of like applied chemistry and also the biological knowledge. So this is also very means the all overall the bioengineering research field is highly related, and also the highly important this biotechnology knowledge is highly important for this application in the bioengineering. So now, like if you take my example, my BTech was in biotechnology. I got the opportunity as a BTech biotechnology to get specialization in my master in biomedical engineering, where I develop biomaterial, and also I for this experiment I again need my cellular culture knowledge, which I got from my biotechnology background. In my PhD level, although I developed some biomaterial, and again I need the application of this biomaterial for the biological system, for doing the cell culture and the drug delivery study. That knowledge also I acquire from my BTech level. And in postdoc now I'm doing the experiment in the animal model. I'm developing some navigator molecule, which is the basically the which I need the molecular biology techniques, and also the cellular culture experiment, and also like the means that totally the animal cell culture experiment. Then those experiments I need the knowledge of biotechnology. So this biotechnology is very important to continue the research in the means basically the bioengineering means it's very important. This biotechnology has a very important role to continue the research in the bioengineering field. And if you see means in every part of the research in this bioengineering, you need this knowledge of this biotechnology. So now I just briefly I want to tell about the research prospect of bioengineering in Japan. So this is basically mainly focused on the material structure, and it's a combination of the research in chemistry and biology, and in the very fundamental level. So in every very fundamental level, you need the knowledge of both chemistry, material science, and also biotechnology. So if you have those kind of knowledge, it's very good to conduct research here. And for that, every lab has excellent laboratory facility for this kind of research. Not only the central facility, each and every lab have all this facility, and it's very good opportunity to continue the research bioengineering research in Japan. And also it is a very research oriented study, like not very means like not, you don't need to study the coursework. It's the main focus is the research here. And also you can get the freedom to perform your research. Like now I'm doing the my research is my own research, and I can select my research area and I can do the research. So there's a complete freedom here. And you can get the fellowship, like is mixed fellowship is already available from Indian student. You can apply there. There are various fellowship here. For every university have their own fellowship. So if you perform well, you will definitely get the fellowship. And now like there is increased availability of the English coursework, because previously when I joined here, there was not much scope, but now almost every university they have arranged their English coursework, so there is no problem of continuing masters and PhD here. So, and thank you very much for all for your time and patience. So, if you have any
Thank
question,
you.
please ask.
Yes, Deep, there is a question. Shubashi sir, we have a question, and that is, what is the basic application of the navigator molecules developed in the lab?
Uh, we are now, this is a new project, so I can't share much, but yes, one of my senior is doing, we have, in his research, that is normally the beta 2 macroglobulin when increased in the biological system. So we just try to make some navigator molecule which have a both targeting point. One is the target point of the capturing of this beta 2 macroglobulin, and another point is to just means just to bypass it, or to just navigate it towards the liver so that it can metabolize, and then the blood level, the beta 2 microglobulin in the blood level can be reduced. So we design a navigator molecule so that it has a both targeting point. One is the capturing site of this protein, and also another is the navigating site, which is has a receptor which has means the in the liver, liver navigating receptors. So in this way we are just trying to reduce the means the decrease the concentration of this beta 2 macroglobulin in the blood stream. But my project is a little bit of different from this, but the overall the navigating system is the same.
Okay, Deep. Now the second question from the student side is, how did you opt for research as a Btec, via GATE or GRE?
So I applied for the next fellowship. So for that I just contacted with my PhD professor, my supervisor, but I didn't get. But he said you can come and we will arrange some fellowship. So I just go and I got the fellowship for whole 3 years, and it's very good amount. It's same as the next fellowship. So if you are interested to do the research, you can write to the professor, and then maybe definitely they will give some reply. And you if you, especially if in the first 6 months, if you perform very well, so you will definitely get some fellowship. And also, like in my case, I got the waiver of my entrance exam and my entrance fees, and also my enrollment fees, and also my tuition fees. So I got the waiver. I have to pay the half of this, so I got this uh, a kind of fellowship along with my fellowship.
Okay, Deep. And then the last question is, how did you opt for fellowship in this institute? Through your professor, or did you? How do you search for this one?
This fellowship is available in the university, so best you have to apply.
Okay. Okay, Dee. Thank you so much. That's all for the questions. Thank you so much, Sushmita Dee.
Thank you so much.
Dee, you can stop sharing.
Okay, okay.
Okay, so moving on to our next speaker for this evening, we have Miss Mithika Roy. She is a scientist at Vantage Research in Chennai. Mithika Dee, are you here? Mm, Mithika Dee, you're not audible. Okay, Mithika Dee is having some technical problems. So, Sneha Dee, are you here?
Yeah, I'm here.
Can you go first?
Yeah, should I
Okay.
start?
We are moving on to Sneha Dee. She is our next speaker. Ms. Sneha Das, she is executive in Biocon Biologics, Bengaluru. Yes, Sneha Dee, you can go now.
I'll just switch on my video 1 second. Am I visible?
Yes, Dee, you are.
One second. Somehow my head is getting cut. One second. Yeah. So, good evening, guys, and very good evening to all the faculty present here. First of all, I would like to thank all of you for inviting me here to give this talk. Um, I'm sorry I don't have a PPT to show you guys. I haven't been keeping well since one day, so I'll just give a brief overview of what the industry is actually. And since you guys are you'll be fresh out of college in some years, and Debanko and some fourth year people are also here, so you can get a new perspective of what the industry actually is, and what we do here. So basically, I am, I'm working in Biocon Biologics. It is a separate entity from Biocon Limited, and we are into manufacturing biologic drugs and biosimilars. And Biocon Limited also manufactures API drugs as well, which are better known as generic drugs. So what these are, I'll let you know in the course of the talk. So just to begin off, I'll give you a brief overview, like I said. So basically, what do you think happens in the industry? I would like to make this an interactive session, if you don't mind. So, what do you think happens in the industry? Like any idea, any sort of idea you guys have, since we are all in the same field, and I'm just inquisitive as to what you actually know. Based on that, I'll modify my lecture here. So, anything, and I would like to know specifically from the students, because I'm pretty sure that our faculty knows everything. So, yeah, anyone? No one? Then I don't mind picking out people.
The you can go on, like, oh, you can share what you have thought of, because I don't think so. My seniors are like, the mics are not on.
Okay, so okay, fine, fair enough. So I'll just move on to my talk then. So basically, the first step of any drug manufacturing is R&D. So in research and development, we actually, what they do, I'm not in R&D, I'm in production, which is the next step of the process. So in R&D, they research about what drug has to be made and how it can be made. When it comes to biosimilars, which are basically similar to novel biologic drugs, they figure out the best way to manufacture the process, and they do it in a small scale in their labs. So after the R&D process is approved, we move on to, the process is entirely scaled up, and the entire process transitions to production, which is the department where I am working in. So in production, there are two types of production: upstream processing or production, and downstream processing and production. Upstream, basically, they extract the material or the API, which is better known as the active pharmaceutical ingredient, and they extract that from the living organism, and no amount of further processing is done in upstream. The rest of that happens in downstream, and I am in the fill finish department of Biocon Biologics, wherein we take the API from the upstream department and we add excipients to it, and we fill it in and finish it off. So basically, I'm in the last stage of the production. So you might get questions in your mind as to what excipients are, what upstream and downstream processing are. So you can just ask me after my talk. So after production, after it gets approval and all, it moves to quality control. So quality control is a allied department. They work closely with us. So when we formulate the drugs, and before filling it in, we have to take approval from quality control department. So in quality control, again, there are two types: quality control analytical and quality control microbiology. So in analytical, they assess the characteristics of the drug product. Basically, by there are various different assays there, and I'm not going into the technicalities of that. You can Google it up anytime. So, and in microbiology also, they do certain types of assays. So quality control is basically controlling the quality. If anything is out of limit, or out of specifications, they inform us, and we have to stop the process midway. So in that case, when a process is getting stopped midway, it is not desirable, it is not recommended. So what we do then, we raise deviations. So that is the main department where I'm working in. I am in deviation management and change controls also. So in deviation, what we do is we assess as to why it has happened, because at the end of the day, it is creating an impact on the quality of the drug product, and this drug product will eventually go out into the markets, and eventually people will buy it. So we don't want to compromise on the quality, right? So what we do is we assess as to why this has happened, and anything which is out of normal, or we basically operate on a list of procedures or a set of standard protocol, which we know as the standard operating procedures, or the SOPs. So if anything happens beyond the SOP clauses, we raise a deviation. There is a specific procedure for raising a deviation. So we raise that. Um, we investigate as to why it has happened. We try and go to the root cause as to why it has happened, and to prevent it from happening further down the line, or at a later stage. We take change controls. So in change controls, what we do is we recommend kappa. Kappa is nothing but corrective action and preventive action. So as an immediate step, to rectify the mistake or rectify the discrepancy that has happened, we actually do a corrective sort of action depending upon the mistake, depending upon what has actually happened. And to prevent it from happening further, we do a preventive action. So that, together they are known as kappa, or corrective action, preventive action. And then again, we have a specific procedure for going about that change control and kappa. So if in case you are interested to know further, you can again ask me at the end of the session. So after that, are these okay, Devanko? I'll take up your question after my talk. So after production, after quality control, the entire process is presided over by the quality assurance department. Most of you will get confused between quality control and quality assurance. Both are kind of similar, but not exactly the same at the same time. Quality assurance, they assure the quality. They see to the entire process, and we have different sectors of quality assurance as well. We have in process quality assurance, we have sterility quality assurance. So they are engaged in different functions of the process. So in process quality assurance, they make sure that we are not deviating any sort of SOP clauses during the process. And sterility guys, they assure that the sterility or the quality, again, at the end of the day, of that drug product is maintained. So after quality assurance, we have the regulatory affairs department. Um, and finally, the drug goes into the clinical trial stage, and finally the market. So the clinical trial stage is of course applicable for the novel biologics that we manufacture. For the biosimilars or the biologics that are already approved, we don't have that clinical trial stage, and it moves directly into the market after after regulatory approval. So that is basically the entire flow process of what we do in the industry. And you guys, I'm just telling you this because you guys are just fresh out of college, and you must, before entering the industry, you must get an overview. Because personally speaking, there is a huge gap between what we study in college or what we study in school, what the idea that we have of biotechnology, that is entirely different from what is happening in the industry. So I was lucky to join Biocon Academy also and get a entire understanding of that process, and I'm just passing on my limited amount of knowledge to you guys. So that will be all, and I would be happy to take up your questions now, if any.
Niti, can you go for the Bankada's question first? Like,
Yeah,
uh,
yeah.
these deviations amended into the set of protocols for further downstream processing?
Yeah, these deviations. So once a particular deviation happens, we try and amend that as to, like I said, so that it doesn't happen further down the line. And we include that deviation in the change control so that we can amend our SOPs also. So based on our deviations, correct, they fall under CAPA. That will be CAPA, CAPA, corrective action and preventive action. So we take it into consideration under CAPA, and we make the necessary amendments in our change controls. And finally, the final step will be the revision of our standard operating procedures so that it doesn't happen again. Like, did you like get what I tried to say?
Yes, Deep, perfectly fine. Thank you, Deep.
Correct. Thank you. Any
He
other?
wrote another question that how did you get the job after BTech in Biocon? Like, what is the
Okay.
career that you went through?
Yeah. So basically, I was with Infosys for a brief period of time. After I completed my BTech, I got placed there. So 6 months I was there, and it was basically a hell and heaven sort of a difference for me. Coming from a biotech background and transitioning into an IT background was completely different. So I left that job and I came back home and I applied to Biocon Academy. So these people, they provide you with that knowledge, and they there are several sorts of courses and programs which are available there. It is also based in Bangalore. So yeah, I got there and I studied for a period of 4 months there. I underwent a training. After that, I directly got placed into Biocon.
Okay, Dee. That's all for the questions now. Thank you so much, Dee.
Yeah, thank you.
Okay, so moving on to the final speaker of the evening, we have Ms. Mithika Roy. She is a scientist at Vantage Research, Chennai. Mithika Di?
Yeah, hi. So good evening all. And thank you, Ishna, for the introduction. And before I start my presentation, I would like to acknowledge and extend my thanks to the professors and the teachers present here who invited me to share my experiences after I graduated from Haldia Institute of Technology. And it's a great feeling being here. I know the theme of today's talk is the prospects of biotechnology in the post-COVID era, and I am also aware that most of the esteemed speakers who have previously spoken before me, they have discussed what are the avenues which are available to the current students in order to get a fulfilling career in biotechnology, either in the academic field or in the industrial field. And that has already been covered in a more general and broader sense by the speakers before me. So I would not like to delve into a lot of details regarding that, but instead take a moment to actually make you guys aware of what I do as a scientist in Vantage Research. And before that, okay, sorry, let me share my screen. Is it visible?
Yes, Di, it is.
Yeah. Yeah. So before that,
Yes, Deep, visible.
thank you. Yeah. So before that, a brief introduction into my background. I completed my Btec in Biotechnology from Haldia in 2014, and after that, I cracked GATE and I pursued Mtech in Biomedical Engineering in IIT Bombay for 2 years. There, I had for my Mtech thesis, I did work on a drug delivery system to deliver antibiotics into patients who were suffering from drug-resistant disease. But after that, due to campus placements, I landed my current job, which is as a scientist, specifically biologist in Vantage Research. A little bit about Vantage Research. So it's a startup which is based in Chennai, and it was established in 2013. And this is a mathematical modeling company, but we do a very niche kind of modeling, which is called quantitative systems pharmacology modeling. And in this way, we help assist the research and development process that happens in the pharma industry. Most of our clients are big pharma, like Eli Lilly, Merck, Genentech, which are based in US, and also MTPC, which is like Mitsubishi, which is based in Japan. So these are our clients. Coming to our team, it's a very diverse team of engineers and biologists. By engineers, I mean people who have done their masters or bachelors in mechanical engineering, production engineering, civil, aeronautical, etc., as well as people who have basic science degrees in physics and applied maths. And for biologists, people such as me who have done Mtech in biomedical engineering, who have pursued basic biology research in cancer biology. So it's a very diverse interdisciplinary field where engineers and biologists work hand in hand to build models of disease areas. And we basically work in the domain of systems biology. So this kind of modeling approach that we do will be mostly inclined to people who really like who are aligned towards maths, physics, as well as who really like coding a lot. So when we build our models, we basically code in MATLAB, Python, R, etc. And also because the model will generate biological results, a bit of biological knowledge helps. So this is a bit about Vantage Research. Now I would like to delve a little deep into what kind of modeling we do. But before that, I want to describe why modeling is required, especially in a pharma industry. And for that, as you guys are all aware, that the drug development process is a very intensive process where the pharma industry invests billions of dollars. It takes almost 10 to 12 years, right from the inception of a drug, from identifying a lead molecule, and then the pharma industry has to, or our clients has to conduct all the 3 phases of clinical trials before applying for an approval from the US FDA, which is the Food and Drug Administration based in USA. And only if FDA sees good results from the clinical trials, they'll approve, and then the drug will be marketed. So it's a very time-consuming and intensive process. It's a very trial and error process. It's an iterative process, and since it's a very time-consuming and a lot of money goes into it, and it may happen that any stage of the drug development process, the results might look that it is not good, or the drug is not efficacious, or it has adverse events to that is happening to a certain group of patients. That will be very disruptive. So in this scenario, the modeling generally helps if the pharma company, who are generally our clients, they get a heads up on the direction that the drug is going. So if they have a new drug and they generally come to us with a question like whether the new drug that they have is better, or at least at par with the standard of care that is available out there. So if it's a cancer drug, they would ask whether the new drug that they have is better than the chemotherapy drug that is suggested to everyone. or the drug that they have, if they have a certain kind of adverse events in a particular group of population. So these are the questions that the pharma industries deal with, and they come to us so that through computer modeling of the disease, we can help them answer these questions. So that's about it, about why modeling is required. And the modeling that we do in our company is something called Quantitative Systems Pharmacology Modeling, or QSP. And that involves a mechanistic understanding of the disease, the complete disease physiology. So what we do is we uh, go to the literature and we read a lot, we gather a lot of data on the scientific evidence that is out there about how the disease actually initiates in a particular patient, how it progresses, the drug that is targeted towards the disease, and if a drug is failing, why is it? So all this information is available in the internet out there, based on experiments that have been conducted, based on clinical trials that have been conducted, and we try and put all this information into our model. So basically, it's a knowledge integration that happens right from the cellular and the tissue level, and it goes all the way up to the population level. So we know what are the perturbations that are happening in the tissue level or in the cellular level, what kind of interactions that are happening between different cells that is actually leading to the manifestation of the disease in the population. And that also explains why a particular patient or why a particular group of people with a particular disease, say cancer, responds to chemotherapy. And if they are not responding, why that may be the case. So this is a little bit about the QSP process, and the QSP process basically has six stages, and I will be going a little bit deeper into what are the six stages of the QSP process. So the first stage is basically on identifying the project goals and the needs. The first two stages, I'll combine it and relay what actually happens in the first two stages. So throughout this talk, I will be giving the example of cancer in a very, very simplified way. So if our client has a new cancer drug, which is not a chemotherapy, but which is something called an immune checkpoint inhibitor drug. So if they have that drug, they will come and ask us the question of whether it will be better than the chemotherapy which is already available in the market, and whether it will be effective, whether it will be efficacious to a person who is suffering from lung cancer. So this is the question that they come to us with, and for that we have to first identify what kind of model we want to build. So since it's a cancer drug, we know that we have to build a cancer model. But as you know, all cancers are different. So if it is targeting only lung cancer, we'll focus only on a particular kind of lung cancer. We have to prioritize what kind of model we want to build. So if it's a non-small cell lung cancer model, we can only address questions related to it and not small cell lung cancer. So this is the part in the stage 1 and stage 2 where we define the scope of the model. Here is the stage where we make a lot of assumptions. We say that we can only model the lung cancer, and we'll assume that the patient doesn't have any other underlying condition which might interfere with the cancer. So this is the stage where there's a lot of hypothesis, where a lot of assumptions happen. This is the stage where we actually determine what is the scope of the model. Then coming to the stage 3 is where we represent the biology of the system. So what we do here is that this is the stage where actually my understanding of the biology comes, and here is the stage where the subjects that I studied in Btec in Biotechnology at Haldia generally helps me a lot. So here I go into the literature, I go into the internet, and I try to understand as much as possible about lung cancer, why it happens, what are the key mutations that defines the cancer, how it initiates, how it progresses, what are the key therapies that are available to tackle lung cancer, etc. So after this knowledge integration happens, what I do, and before that, I would like to say that the knowledge of immunology, the knowledge of biochemistry, microbiology, as well as RDNA technology, and also animal technology, really helps me in this effort. Because in order to understand the experimental papers that have been the animal studies and in vitro experiments that have been done to understand lung cancer, that requires the knowledge, the basic knowledge of the subjects that you have studied in biotechnology. So after this thing is, after the knowledge integration happens, after I understand a lot about the lung cancer biology, I convey this information to the engineers, and the engineers represent the engineer's task is now to represent the lung cancer physiology in a model. By that, I mean they create something which is called a model map. So I have identified that if it's a lung cancer model, the key players will be tumor cells, the key players will be different kind of T cells, B cells, and the cytokines that interact between them, the ligand receptor interaction that happens between them. So once I have established that, the engineer's job is to represent each and every player through equations, through mathematical equations. It can be ordinary differential equations, it can be partial differential equations, etc. So each set of model species will have its own set of equations. So the tumor cell will be represented by a proliferation rate. They will have its own apoptosis rate. They will have an equation representing that if a CD8 cell is attacking the tumor, then the tumor cells will be reduced by this percentage. So once that data is put into the model, let me take a step back. So once the engineers actually, hello? Hello?
Yes, you're audible.
Yes, you are audible. Please go on. Please.
Once the engineers are actually had made this model. Oh, 1 second, sir. I am having a little bit of disturbance. Just give me a minute. Yeah, I'm sorry for the inconvenience. So once the engineers have already made this kind of model, I again go into the literature and try to find the parameters that will represent the tumor cell. So as it is a biological system, we know that irrespective of how much nutrients is available, how much angiogenesis has happened, tumor cells can only grow to a certain extent. So I go and find the data that supports this hypothesis. If it's a lung cancer model, as I said before, we know this is the average size of a lung tumor. This is the density of a tumor cells that is present in a particular biopsy. This is the density of the immune cells that are present in a particular biopsy. So once that data is incorporated into the model, this incorporation of the data actually makes the model more robust, and it makes it biologically feasible. Because if we don't increase our confidence in the model by putting the biological data, it can throw any result, and which is not biologically feasible, and that might lead to wrong results when it is trying to predict what will happen with the drug of the clients. So once this model map is made, and once all these parameters representing the tumor cell, representing the CD8 cells have been put into the model, again, what I do is I find the relevant clinical trials that have targeted lung. So these kind of trials are available in a site called clinicaltrials.gov, and there it's a clinical trial registry, and I find the relevant trials which actually led to the approval of the standard of care treatments. So it can be either a chemotherapy drug, different kinds of chemotherapy drug, chemotherapy cocktails, or it can be something which we called as immune checkpoint inhibitors. So once we identify the clinical trials which have been instrumental in an approval of a chemotherapy drug, we read that paper thoroughly. We try to extract as much as possible information as well as data from the clinical trial, and we train our model using the data and the information that has been put in that has been extracted from the clinical trial. By that I mean we create something which we called a reference virtual patient. And by reference virtual patient also I mean that we built a model replica of a real life patient who has a certain baseline characteristics. And these are the baseline characteristics that we generally see in a clinical trial information. As you know, there are a lot of people who come into the trial who might have different tumor size, who might have different hemoglobin level, who will be of a certain weight, who will have different density of immune cells, or who will have different CRP level. And we try to create a model replica of that patient who has come into the clinical trial. And then we give the chemotherapy drug that is used in the clinical trial as an input to the model, and we ask the model that if the input is this drug, if this drug is given as an input to a reference virtual patient, then the output should be this. And the output is basically what the clinical trial result that we see. So in this way, we actually train the model again and again. It's basically a little bit like machine learning, where we train the model using clinical trial data. And if you incorporate more and more different type of therapies in the model, the robustness of the model increases, the reliability of the model prediction increases. So this is the stage where we do a model. This is the stage where we do a model calibration. Basically, we train the model using the clinical trial data. Once that happens, what we do is we move from a reference virtual patient to something which is called a reference virtual population. And by virtual population, again, we mean, as you know, that different people who come into the trial, they have different variabilities depending on their DNA mutations, their biomarker level, the tumor that they have may be of different size. Some may have metastasized, some may have not. So we try to replicate the population that have come into the clinical trial in the model, because that would increase our confidence in the model. So once we get to this stage, so if you see here, this may be a reference virtual patient where and from here we actually we change the parameters, we change different parameters to actually get to this level, which is called a virtual population. So once this phase, which is called calibration, that is done, we move to something called validation. So calibration stage was where like the input and the outputs were defined. We were training the model using the data obtained from the clinical trial. But when it's a validation phase, we take a clinical trial and the model has not been trained with the data set before. It's a very novel data that it is first seeing. So we give the input to the model, and we see whether the result that the model is generating, the model output, actually is similar or at least at a close range to what the actual trial output suggests. If it is similar to what has happened in the trial, then well and good, our model is robust, our model is capable of making accurate predictions. But if it's not, then we go back and we say, like, why is this the case? We ask the question, why is it that our model is not able to predict, not able to give the results that we see in real life? And then again we have to, it's an iterative process where we go and we have to change different parameters, or we figure out whether we have missed a key piece of biology which is hampering with the predictions. So once the validation phase is done and we have sorted the issues or something, then only we give the novel drug which our client has, and we allow our model to make predictions, and our model generally gives results like efficacy. So if a company says it has a new drug, so we give it as an input and a model predicts if it's a cancer drug. If that drug is given to the model, by how much percentage the tumor will reduce. if it has any side effects, also if there's a certain number of people who will be completely resistant to the drug. So these are the questions that we try to address by QSP. There can be other questions also, which is far from efficacy and adverse events. Sometimes our clients already has a clinical trial result data, and they will ask us to interpret why is it that we are seeing the results. So then again, we go to the model and we try to understand what are the perturbations that are happening in the biological pathway, whether there's some cells which are actually the culprit, which is not giving us or like giving the client their desired objective. So these are the kind of questions that can be addressed by QSP modeling, and it's to people, I would say, and especially the current batch, I would like to say that it's not just QSP. There's a lot of scope for network modeling as well as statistical modeling, but they are very, very driven by data. However, the modeling that we do is more of a predictive kind of modeling. But since COVID, you can and as you know, the drug development process, because it takes such a long time, the time scale at which new drugs has to be out in the market has been reduced, and in this way the modeling can definitely support the clinical trial results. It will I am not trying to undermine the effort of the clinical trials, and we know that FDA will definitely not approve a drug without actually having clinical trial. But in this post-COVID era, I feel modeling, not just QSP modeling, any kind of modeling will be a great support system that actually emphasizes and explains the data that we see in clinical trial. So instead of waiting for like years, 2 years, 3 years to actually get the result and then analyze, you can definitely rely on modeling to at least get a heads up on where a key drug is going. So that will be all. Thank you for your patient listening, and I'll be happy to take questions.
Thank you so much, Steve. That was really an interesting session. And we have some questions for you now. Well, the first one is how to effectively prepare for GATE exams, the do's and don'ts that you followed and you would recommend.
I would say that the textbooks that we have are generally sufficient, but I generally relied on most of the textbooks that were suggested by I think WBOT at that time, and it was sufficient. But I already also bought a book which was mostly on objective questions, so that at least gives you an idea about what are the key areas where GATE focuses. But yeah, that's about it. And also I would say that please prepare well on the mathematics section, because sometimes even if you are very, very strong in the biology section, if you don't have that cutoff for the maths, then that's a problem. So as biologists, we generally tend to ignore the maths, but please pay focus on the differential equations, permutation, combination, etc.
Okay. Thank you, Dee. And then the next question is, what are the research scopes in the field of biomedical engineering? Could you share anything?
Research scope in
biomedical engineering.
Okay, so biomedical engineering is, because it's an interdisciplinary field. You can either go to you can either be more biologically oriented or instrumentation oriented. So if you are from the biotechnology background, you can help in tissue engineering. You can help in developing biocompatible drug delivery system. The project that I did. You can help in making prosthetics and all. That is all the novel areas that are in the biomedical field. But in case people from instrumentation background, they generally try to focus on developing point of care devices. That is their strong suit. But yeah, the tissue engineering field as well as the drug delivery system, I think that is the main. That is the key novel area from biomedical.
Okay. The next question would be, uh, does vaccine preparation also need a model map? And how is it different from drug modeling?
No, so we generally don't do vaccine modeling because vaccine is something that is given to a healthy patient. What we focus is all disease area which are already established disease and which has drugs for it. Since vaccine depends a lot on whether a healthy immune system will mount, whether a healthy person will mount an immune response, there's a lot of variability and a lot of uncertainty in vaccine. But I would say that the established disease area generally doesn't have that much uncertainty. So at Vantage, we are focused on developing models for diabetes, psoriasis, cancer, etc. But we are not into vaccine models.
Okay. And the last question is, can you give us a brief idea about ligand-based drug designing?
Ligand-based drug designing.
Yes.
Sorry, I don't think I would be the right person to actually know this because we generally don't do the drug designing part, which involves a lot of pharmacology and development of lead molecules and etc. But our clients actually already has a drug in their hand. They have already designed it, and we just say whether that drug will be efficacious, whether it will be effective or not. So I don't think I have the right know-how to answer that question.
Okay. And there's another last question, which is, what is the scope of biomaterial in biomedical engineering?
Uh, the scope is a lot. So coming to
And also the scope of bioinformatics, if you could stress on.
no, so let me talk about biomaterials because I think I did my MTech project in that area. There we were trying to develop a gelatin-based particle which will actually go and release, and the antibiotics will be encapsulated into that gelatin-based particle. And it was basically a targeted delivery where only at a certain pH it will break open. The gelatin particle will break open and release the antibiotics. So the scope is a lot because in IIT Bombay there's a lab. It's of Rinty Banerjee lab which actually deals with biomaterials, and they have different kind of they have developed different kind of liposomes, different kind of gelatin-based particles, which actually is towards the targeted delivery of drugs.
Okay. Shubhashi sir has some questions. Sir, you can just ask them.
Hello, Mitika, can you hear me?
Yes, sir, I can hear you.
Actually, it's a very wonderful presentation. Very good presentation, actually, I should say. That's very informative, and your presentation was is, some slides, but they're very informative. On first thing, I want to ask you that the modeling you are using with only the MATLAB and R software, or is there any other softwares dedicated? If you want to share.
So most of the modeling that we do is on Symbology, which comes as a package to MATLAB. It is a modeling package that comes with MATLAB. So mostly it's Symbology or R. Other than we don't have it's R or Python. We don't have a proprietary software as such.
Okay. And in MATLAB also you are doing the modeling, huh?
So MATLAB comes with something called Symbology. That is where we build our model.
Okay. And another one thing is that you should you just focus on some on the Btec Biotech for the Btec Biotech students, that how do they prepare for this software packages, this modeling, because the company you are working with is a very good startup company, I think. So is there a scope after Btec students, after completion of Btec? Is there any scope then what are the prerequisites for the Btecs, if there is any scope for the Btecs?
So, sir, I am not very aware whether they would take Btec, but the thing is, Yeah, so the thing is, like, we have to go through an interview process, but I definitely don't do the modeling part. I have joined based on my biotechnology and biomedical know-how. So, but I'm sure that like if that we'll have a scope for like interview if there are interested candidates.
Okay. Otherwise, a very good presentation, and your company is doing very great. As a startup, it is doing very great. Okay. Thank you.
Thank you, sir. Thank you so much.
Okay, Mitradi, there's another question from you from Ovijit sir. Do you think such modeling strategy will bring down clinical trial time in biologics, specifically biosimilar space?
So, sir, I really do feel that it might bring down the time taken for the overall clinical trials to be conducted. So, what I feel is that what I have seen is that the companies will definitely carry on with the clinical trial, but based on the modeling results or the predictions that our model gives, they might change a little bit of strategy. So, if we say that through the modeling that this dose of biologic or is not working in the people, then what they can do is that they can have like different arms in the clinical trial where they can give different doses and they can actually see whether that is the case or not. So it's very I don't think they will scrap the clinical trial or go into a different direction altogether based on modeling, but they can try different doses, or they can choose a particular set of patients. If we feel that if the modeling results say that it will only work in a particular kind of group, then based on our analysis, they can try to recruit those kind of groups.
Yeah, thank. Hello.
Yes, sir.
Yeah, thanks, Smritika. Nice answer, beautifully you explained. So, a curious question, since you know biologics being a very complex, large molecule. So, and you see that one of the major challenge why biologics is slogging is the time taking 7 to 8 years, 5 to 6 years. So, as you said, there are a couple of modeling strategies that you had mentioned in your earlier part of the slides where you can actually predict different strategies which could probably bring down. So, as you said, that you are currently working with different clients like Mark and a couple of other international companies. So, are they working on the pharma space, or they are also trying to bring down the time of clinical trials for biologic space also?
So they are basically working in the pharma space, sir. They are working in this pharma space. They have already clinical trials ongoing, but as you know, the clinical trials they will take at least like 2-3 years. If it's a phase 3 trial, it will take 3-4 years to actually the results to come in. But if they want a heads up, at least like an idea about where the drug is heading, they generally come to us with a question that these are the PK/PD characteristics of the drug. Can you just like model it and see whether the drug will be efficacious, whether the drug will have adverse events, and based on the model predictions, they we inform them to our clients, and based on that, they generally take the decision. At least they are aware that where the drug is headed.
Yeah, you're right. Actually, in my opinion, is like, see, for an example, novel biologics is your footprint is new, but in the biologics, when you talk of biosimilars, I think this is a potential strategy what a lot of companies should investigate to bring down the time by incorporating this particular thing. So I wanted to know like your thoughts on that. I mean, that's my opinion. Since you're working in this field, and I potentially see that, especially in the biosimilar business, there will be a lot of area where this can bring down the overall turnaround time for trials and all this thing.
So, sir, thank you for the question. I'm not very aware of the biosimilars because generally the companies that we work with, they already have like a new product, and they generally want to compare the efficacy between the standard of care which is already established in the market and the new thing that they have. Because these are like huge pharma giants, I don't know whether they are into manufacturing biosimilars.
Yeah. Otherwise, your talk was very nice. It's a wonderful talk, Mritika. Thank you.
Thank you, sir. Thank you. I'm really glad that you guys could understand. I mean, I'm not very sure whether like it was a very technical. Because I wanted to make it a little bit light for the students, because even as I said before, this is also a very new area for me. The modeling space, especially in India, only a handful of companies does it, and the modeling that we do, the QSP modeling, that only mostly like Vantage Research does it. So even I had to work my way up and learn a lot in the process. And as Sneha said, your knowledge that you get from biotechnology, the subjects that we study in biotechnology will always be helpful. But in the industry, it's a completely different ball game, where you have actually learned different ways and strategies to incorporate the biological knowledge and find its application.
Yes, absolutely right. I also think personally there are a lot of potential hidden in this area, which especially in India, not much has been explored earlier. And a lot of students should have opportunity who has keen to do something in line with mathematics and biology, a combination. This is probably a new area which are picking up. That I also think so.
Yeah.
And by the way, a quick intro. So I was the first batch from HIT 2005 batch. I've been in this biologics, especially
हाँ
drug
सर आपने कॉल
development,
करना
bioassay field last 15 years. All right. Thank you, Mritika. I will hand over to the organizers.
Okay, Dee. So it was a wonderful presentation, and I hope that all the students who are watching right now in the stream in the YouTube via the live session have fulfilled their queries. We'll wait on for just a few more minutes and a few more seconds, and then we will go on with our vote of thanks.
Didi, that was really an interesting session that you gave us, and quite insightful on your work. And I would, uh, well, since you cracked GATE and you got it in one of the most prestigious colleges in the country, uh, can you share us about your experience and journey? Didi, are you there?
Yeah. Ishna, is the question directed to me?
Yes, of course.
So can you repeat what was my experience?
Well, you cracked GATE quite successfully with an All India rank of 25, I guess.
Yeah.
Yeah, so well, congratulations for that, and that is really motivating. So, if you could share how you prepared and how was your experience there?
Okay. So the thing is, like as I said before, for GATE, I mostly relied on my textbooks. That is like the most comprehensive. If you are well aware of like all the things that are in the textbook, I think it is well enough for good enough for GATE. But as I said before, also maths plays like a very important role. I attempted GATE a second time, but like my math score was actually like not very good, and for that, like I think like yeah, I did not score get the rank as much as before. So definitely don't ignore the maths section. I would say this is my opinion completely, and focus on key areas like you will always know by looking at different papers of the previous years that they target a particular kind of subjects. They target a particular area in every year, and it's actually repetitive, so it might give you an idea about like which are the subjects which you really need to focus on. Immunology, there's a lot of questions on immunology and biochemistry. I feel, at least like from my opinion of giving GATE wise. So definitely focus on the key subjects and maths, that I would say. Also, after cracking GATE, I got into I got into IITs. There also you have to choose whether you want to pursue with biotechnology or biomedical engineering. I switched to biomedical engineering because then at that time I was the need for me in my scenario, the need of the hour was to get good placement, and I felt that if I work in a biomedical field which is very application oriented, probably my chances there would be better, and that is why I chose IIT Bombay. After coming into IIT Bombay, I felt that so here is what I would say, and this is my experience. Biomedical engineering is already a Btec course, and people who come from biotechnology background and they have to study biomedical engineering, it's a little bit difficult for them. At least I had a really hard time, because you have 1 year for the subjects to cover like the entire course, and 1 year to focus on your MTech project. So there, because you have half semester courses also where they teach a lot of instrumentation, a lot of physics, a lot of medical imaging background, the kind of subjects. And because I was from a biotechnology field and I have already mostly focused on the basic biology, I felt that part of the journey was a little difficult, because people who come from the instrumentation background, electronics background, who come from basic physics, they are definitely ahead in days, because they know their electronic engineering, they know their thermodynamics process. So that was a bit difficult, and I don't mean to scare or deter any of you from taking biomedical field, but because it's a half semester course where you have to know a lot within 3 months and write an exam, I feel that people who come from the biotechnology and they have to tackle all these very heavy electrical and electronic subjects, they are at a little bit of disadvantage. If you can do it like well and good, and I really like you to go with it. But these are the challenges that I faced with especially these subjects. But then the good part about being in IIT Bombay is that you can pick and choose your subjects except the core subjects, which are like electrical, which are physics and all. After that, you can definitely, if you are more biology oriented, you can go for protein engineering, you can go for genetic engineering, etc. And there are a lot of options and avenues available, because you can pick and choose your subject based on your interest. You can even do a stats course. You can take a if you just want to do a English literature course, you can take it and take that will help you in making credits. So that is like one of the wonderful things of the being in IIT, where you can choose your subjects except the core subjects. And the next year, the second year, basically goes in research, where first you have to in consultation with your guide, you have to choose your research subject, and there you can do your research in a kind of any field. There are many people who were in my batch who actually did a project in electrical engineering field. So there you can, as long as you can have a little bit of like biological relevance, you can do your research project in any department and no one says anything. So that is like one of the very liberating and very good thing about IIT Bombay, that once you tackle the core subjects, once you get passed through the hurdle of the core subjects, you can pick and choose your subjects as well as do research in any field of your choosing.
Okay, thank you so much, Steve. With that, I would like to conclude and come to an end of this session. It was really wonderful to have all the three speakers with us share their knowledge and their experience. Thank you so much. And now I would like Suraj sir to give a vote of thanks.
Thank you, Isna. I, Professor Siraj Datto, on behalf of Department of Biotechnology, Haldia, Institute of Technology, Haldia, the entire supporting team of this webinar series, and my own behalf, extend the heartiest vote of thanks to all the speakers for sharing your opinions on this day. A big thank you to Dr. Suchismita Chatterjee, our ex-student and fellow researcher at National Cerebral and Cardiovascular Center Research Institute, Japan.
Thank you, sir.
Hello?
Thank you, sir.
Okay. For sharing the story of her journey in this field. I must convey our deepest sense of appreciation to Mitika Roy, who is also our ex-student and a scientist at Vantage Research Chennai. I wonder how a shy, studious girl has transformed into an industrial trainer. Thank you for sharing your experience.
Thank you, sir. It's a pleasure.
Last but not the least, I must thank my dearest student, Sneha Das, an executive at Biocon Biologies, Bangalore, for sharing her valuable experience in this field. Thank you, Sneha.
Thank you, sir. Thank you.
I apologize that I cannot take everyone enough for this, their involvement and willingness to take part at, and extended their helping hand to organize this webinar series beyond this comfort zone. I finally would like to conclude my expressing the cordial thank you to all our audience for being with us in this evening. Your enthusiasm